So when I took the history, okay, I took the 3 generation history, okay, but the patient was like, okay, I don't have anything on my paternal side. It's all, coming from the mother's side. But as we started doing testing, okay, and like after testing, when we did not find anything like any mutation, okay, and then we took a history again to look for it, okay, and then the patient comes saying that no, my paternal aunt also had a history of breast cancer.
Hi, I'm Dr. Anup Rawool, DM Medical Geneticist, working in the capacity of Associate Director in the Department of Medical Genetics at SRL. Next generation sequencing technology has taken the world by storm. You talk of a disorder and there is an application of next generation sequencing. It's a sophisticated technology, not many aspects into it. Some in the lab, some with the computer system analysis and then the reporting. So today let us understand in short how the next generation sequencing actually happens from the lab to the reporting and which are the experts involved in the entire process. So when you talk of next generation sequencing, things starts with getting the sample in the lab, whether it is a blood sample, tissue sample or any other sample. You do the extraction of the DNA, then you do multiple PCR, laboratory preparations and enrichment of that sample, and you then put the prepared sample for next generation sequencing into a next generation sequencing platform. So that could be Illumina, Thermo, Oxford Nanopore, PacBio or numerous new other platforms that are coming up. Once the sequencing is done, then you have data, what you call as NGS data, GBs of it. So we understand in terms of say numbers. So 1 copy of DNA, 3 GB data, double the population of India at present. And we are finding one missing person in that entire population, double the population. So once you have that data, there is a preliminary refinement of the data that happens, which is done by the bioinformatician. Once that is refined, the processed data goes to a genome analyst who then does a study and picks up those particular genomic abnormalities which may be related to the disorder. And all this data collated is put forth to a medical geneticist. As a team, we sit together and then we formulate the report. So let us understand how these 3 unique professions, a genome analyst, a bioinformatician and genetic counselor, work together as a team with me, supporting me to analyze the most sophisticated data that is generated, giving a comprehensive report, making life easier for the clinicians and helping the patients in their journey of disease management. Today we have with us Mr. Vivek Mane, who is working as a senior bioinformatician in SRL Diagnostics Genomics Lab. He has over a decade of experience in NGS data analysis in various capacities. So let us understand how Vivek works, what are his day-to-day activities and how he eases my life as a medical geneticist. Hi Vivek.
Hi, sir.
So tell me, after the wet lab, once data comes to you, how is that data? How you look at it? What you do with it?
Okay, before that, sir, I want to tell you the workflow of the analysis. Here, when we get the raw data from the next generation sequencing machine, after that, that raw data files are the FASTA-Q files. First, we check the raw data by use of the FastQC tool about the QC check, quality check. If reads are okay, reads are above, means they are falling in Q30, then we proceed that reads for the.
You have cutoff for quality.
Yeah, cutoff of the quality. That quality is the Fred scores. Basically, we use the Fred scores for that. After that, we align that data with the human genome reference sequence. After then we go for the variant calling, and after that we annotate them, and then we report that variants for the secondary or tertiary analysis.
Okay, so these are like in primary analysis, you have the data, you check the quality. Whatever data quality is not optimum, you remove it from your analysis.
Yeah, actually, data quality we remove. Actually, in the next sequencing generation machine, it will be generate a huge quantity of the data, right? There are number of base calls are pre-qualified, they are not good scores. Okay, there may be conflict regions will be there, or small segments of the nucleotide bases are there. So that's why we have go for the QC check, QC quality check. In that QC quality, the reads will be not match, or it will be below the conditions or criteria. Then we have to remove all them.
Okay. So that is like in various stages. You check quality, you then align it with the reference genome, then you annotate, and then you would go for variant calling. I hope I'm
Sure,
giving a
definitely
correct sequence, right?
Correct sequence.
So because you are expert in it, yeah. And that is the primary analysis.
Yeah.
And what happens in say secondary or tertiary analysis?
In the secondary, tertiary analysis, we use means after we capture the variants from the after the alignment, then we go for the annotation purpose. Actually, there are so many commercial and free based softwares are available there in market.
Okay. So to simplify it for our general population or common person, so what Vivek does is whatever sophisticated data comes from the NGS instrument, he checks the quality, he matches it with the reference genome assembly that is there, reference DNA that was generated from the Human Genome Project. Then he checks for abnormalities in that DNA. Whatever abnormalities are there, he collects only those, collates it, and then he does further sophisticated analysis on that to pick up what we are searching for. So that he does with the help of various softwares and algorithms. So let us understand these softwares and algorithms from him. So what all say software? So these are freely accessible or subscribed, or you can just tell us?
Actually, both options are open for the analysis, that you can use the commercial software also, and you use the free based softwares also. As per the for the alignment, you use the Bava tools are there, or Bounty tools.
Say for one whole exome sequencing test, so that is the coding part in the DNA. If you're looking at the entire coding region of the DNA, that is 1% of the DNA. How much data is generated for one sample, say one test, one NGS test?
For the one sample, it will around goes to 8 to 9 GB per sample data.
8 to 9 GB, and you're searching for one abnormality, one bit abnormality in that entire 8 to 9 GB. That's huge. That's huge.
Means your 1 GB will be goes to 1000 million base pairs. Okay, means you can searching one base.
So once you are analyzing data to the tune of 9 to 10 GB for one patient, what is the kind of instruments or computer systems that you require?
For that, normal systems not working for the analysis. You have to get a high configuration systems.
So do you need to have specified systems and customize and get it made, or you do it on internet servers then, or cloud based?
No, actually we go for the servers which is customized, updated for us. That will make it easy to handle them, and our data security will be safe in that things.
So just one small question, like whole exome analysis for one person, after you've got the raw data, approximately how much time does it take? Approximate.
Actually, when we go for the element, it will be take 1.5 hours. For variant calling, it will take 2 hours, near about 2 hours. And after annotation, it will be not that much time to take for that.
So this you're talking of one sample.
Yeah.
Provided you have access to the internet servers.
Yeah.
What if you have multiple samples at one time then?
Yeah. Here now technology will be changed. As per that, there is the new Python packages will be developed. Okay. There is a Nextflow, Snakesmake, where we can use them, and we can put it every sample as in container, and at a time you can run that samples in meanwhile time. Means
in real time.
Yeah, real time. Means you have to one sample you take 3 to 4 hours, by the use of SnakeMake or Nextflow, you can run 10 samples in that 3 hours.
So this is a technology, are you referring, that was used in a Stanford study on Oxford nanopore? So real-time sequencing is happening.
Yeah.
At the same time, you're doing the NGS data analysis with reference genome and picking up the variants.
Yeah. But for that you have to need some very high kind configuration servers to do that thing.
Oh, so that is more of a research-based setting. It will be used, I guess, not in diagnostics as of now that I'm aware. Or is it being used?
No, actually, peoples are going to adapt the technologies, because recently you see for the whole exome analysis, some peoples are that promises that they have take a very minimum time for the analysis. Okay, they are doing the same thing, but when you can use that technology, they only provided you the cloud-based.
So you want to say that for real-time analysis for NGS, you should have best of internet service, that is one, best of internet speed,
Yeah.
that is second, and you should have cloud-based analysis subscription to those softwares.
Yeah.
So that is what is required. Yes. 8 to 10 GB of data from one exome sample, doing a primary analysis, secondary and tertiary analysis, and then filtering out and giving those annotated files to the next team, the genome analyst team, and making our lives a bit easier.
One of my friend I happened to talk to, so she introduced me to a medical genetic counselor from Australia. I think she's one of very few who understands microbial related counseling. And it was like around 5-6 years back. She was talking to me. I had no idea what she was talking, because in medical college we don't study this. And then I was talking to Dr. Anup, and he was explaining, and I was like, I don't even remember studying this, because we study genetics hardly for 15 days in first year MBBS and never after that. And you did your genetic counseling, so you are a genetic counselor now. So what do you study basically to become a genetic counselor?
So what I have done is I have done my BSc in biotechnology, and then I went for a specialization, that is MSc in biomedical genetics with genetic counseling.
Okay.
And then I got certified by the board, that is Board of Genetic Counseling India, and now I'm practicing as a genetic counselor.
Oh, beautiful. So with SRL you are working as a genetic counselor.
Yes.
Okay. And what do you do? Like, what's your role actually?
So my role is to give out genetic information in a simplified manner, okay, to patients or their families, okay, who have a risk of transferring or inheriting a genetic condition. So that is my role. And here at SRL, I work with a team, actually an incredible team, okay, of a team of bioinformaticians plus analysts plus a medical geneticist, okay, who guides me throughout the journey of genetic counseling.
But how's the acceptance? Because see, medical genetics as an investigation itself is not very common, and patients are actually taking so much of information, and you are trying to explain it. So what's your experience? Are they like open to understand, or do they have too many queries?
So,
how it works?
We see a variety of patients. Okay, we see patients who are willing to accept what they have, and there are patients who are in that dilemma, who are in that phase of like, okay, it's not any genetic condition, it's nothing that I have. So there are various of patients that we see, and definitely as a genetic counselor, it has, like, as I have gone through the experience of a genetic counselor, I have tried to accept the patient as that patient comes, and that's what helps me to guide them through their journey.
And it's not very easy to accept this particular diagnosis. Could be because there is so much of taboo behind it, by saying that there is no cure once you have the genetic disorder, or it's very difficult, or then the family will have something like that. So do you also counsel the family along with the patient?
Yes, So what we do here is we have a pre-test counseling as well as a post-test counseling.
Okay.
So pre-test counseling is like before the testing is done. So in that we collect all the possible history. Okay, like the history will contain the personal history of the patient. Okay, and even the family history. So we'll take a 3 generation pedigree.
Wow.
Yeah, so that in that we'll look at all the possible family background, like if there is any patient who has had like a previous child with a genetic condition, or their grandparents, they have had any cancer history, so all that is noted down. And with that, then we go forward with counseling. So from that pedigree we understand of what conditions could be there in that particular family, and that's how we then understand whether genetic testing is needed or not. And coming to post test counseling, it is done when you already have a report, already you have genetic testing done for that patient, and that patient is has a variant identified, a mutation identified. So in that case, then we explain to them of what of what gene they have, what mutation is there in that particular gene, and like what condition it resembles, and what other management or treatment perspective can be considered for that patient.
Okay. See, history taking is the most important part of medical, practice. Unfortunately, all the very senior doctors, or we have very few doctors for every specialty in India. We do not have enough doctors, and they do not get time to actually spend and discuss about at least the patient's past history.
Yeah.
You are talking about 3 generation history.
Yeah.
And that you do before the diagnosis.
Yeah.
Okay, and that data goes to your team,
Yes.
and it helps them to correlate with the findings.
Yeah.
And then whatever the findings are there, you discuss with the patient once the report is there.
Yeah.
Okay, and this kind of counseling you have to do with every patient,
Yes, with
pre
every
and post,
patient.
every
Yes.
patient. Okay.
And as you have said, yeah, history is very important, because I'll just share an example.
Please.
So I had a patient where there was a history of breast cancer. So when I took the history, okay, I took the 3 generation history, okay, but the patient was like, okay, I don't have anything on my paternal side. It's all coming from the mother's side. But as we started doing testing, okay, and like after testing, when we did not find anything like any mutation, okay, and then we took a history again to look for it, okay, and then the patient comes saying that no, my paternal aunt also had a history of breast cancer. So history is really very important.
True.
Okay, and there have been cases that we are trying to find something, but then later on with extra information we get
Okay.
we get to that result.
Beautiful. And what's the biggest challenge that you face while doing the medical counseling?
Biggest challenge. So yeah, as I said that there are some patients who are easy to handle, like as we counsel them, they are quite what do you say, they are ready to accept it. But some patients are there who are very adamant, okay, and sometimes, yeah, the biggest hurdle is taking history, okay, because sometimes they give that information, sometimes they do not give that information thinking it is not needed. But actually they do not understand that even the slightest or the smallest bit of info is important in genetics.
Correct.
So yeah, that's a hurdle. Those are the hurdles that I face.
Okay.
Yeah.
And since how many years you're practicing?
So I have been practicing as a counselor for almost 5 years now.
Okay. And do you see change in more number of people going for genetic counseling or diagnostics per se?
Yes, because there is quite a awareness now, and we in India are also like expanding when it comes to genetic counselors. So I do see the difference. I see like what it was 5 years back when I was training. It has quite changed. I can say. Yeah. So we need more counselors. We need more counselors to go out there and spread the awareness.
Beautiful. And what's the most fulfilling part of your job?
I actually feel I'm very blessed enough to be in this profession, because it lets me, put myself in somebody else's shoe for at least a moment. And that is the best thing that I have ever experienced in my life. So yeah.
Beautiful.
Hi, Riva.
Hello, sir.
So, you're working in SRL, right? You're working in a position of genome analyst.
Yes.
So, not many people, or rather doctors, not many people are aware what this profession of genome analyst is and what exactly they do. So, let us help people understand. I'll request you if you can just share your insights on this profession and just help us understand the nature of work that you do.
Okay. Hi. So I work in SRL as senior genome analyst. So what I do phenotype specific search for NGS data. So whatever patient comes to us, we check that data, we analyze entire case. So suppose if I have got suppose if a patient has like hypotonia or global developmental delay. So those are my key terms for analysis in this case. So I'll search those terms, I'll get what are the genes associated with it, I'll do a search what are variants I'm getting. So suppose if I'm getting like around 100 to 200 variants, I need to narrow it down to 1 or 2. So to narrow it down to 1 or 2, there are multiple steps involved in it. First, I'll do, there are certain cutoffs and criteria which the variant needs to meet. So like there are in silico tools, there are disease databases, there are population databases, all these things I'll check. If the variants are matching these criteria, then it goes to next step. In the next step, I'll check whether the variant is matching the phenotype or not perfectly. If it is, then I'll follow ACMG guidelines to particularly classify the variant as pathogenic, likely pathogenic, and VUS. If after all the criteria are met, finally we report the variant and the final report goes to the patient.
So just to sum it up, like you have analysis that goes on at multiple levels.
Yeah.
Once you do a next generation sequencing, that is a sophisticated analysis of the DNA, primary analysis, secondary analysis, and tertiary analysis. So primary analysis is what Vivek does for us, bioinformatics. Secondary is refining, that also Vivek takes care. And tertiary analysis, that is picking out those particular variants or genetic abnormalities using sophisticated algorithms, softwares, and checking that it is matching with the disease details that are provided by the clinician, and finalizing or helping me, a medical genetist, finalize the reports, is it? How is a normal working day for you at SRL as a genome analyst? If you can just explain.
So the day would look like, I'll come, I'll check what are the queries I have from the client.
So it will start with how many reports are up for reporting, how many patients data is there with you, right? You'll start looking up with the abnormalities in each report. So on an average, how much time you'll get? So the data that you get, it is in what format? Excel sheet or like, that is one format I know, but if you can explain other formats.
So it generally the data from bioinformatics team which we get is VCF file. So that is called variant calling file. That file contains most of the variants. So we have some softwares where we can upload this file. There are multiple paid softwares, there are free softwares as well. So we have one licensed software for us, so we upload this VCF file there.
So by variants, you mean the genetic abnormalities?
Yes, mutations.
Okay. And you have all these algorithms which you use, open and paid, subscribe for your analysis.
Yes,
right?
Yes.
So on an average, in one, say a whole exome report, how many abnormalities you come across?
So
on an average, without the clinical history.
It will be around 30,000. After once I upload this variant
on an average, say 30 to 40,000.
Yes.
Okay.
Yes.
And once you put in various filters, then you narrow it down to?
It will be around 200 to 300 to max 500.
So that's a huge number. 200 to 300 variants, each variant correlating to various disorders, which one to point, and you use various parameters to look into it, right? So I really thank you for making my job so easier.
But why you went for this particular specialty or this profession?
So NGS is a very recent technology, okay, which is like booming in India and also in the world. I really wanted to help patient because I understand my job profile, what I am doing. I'm literally like finding variant which on the other side patient must be waiting for the report because they don't know as of now that this thing has happened. They can see the symptoms only. They don't have anything molecular report on their hand that it has happened. So I know my job and I take it like really seriously if I have got symptoms or patient details that these are the things like Ria, she gets all the details from the patient. So once she gets it, it comes to me. Now I'll read all those details and everything. I'll check whether there are mutations or not. So sometimes it also happens that this test is not able to pick it up. Okay, there are other tests. But still, I have to give my effort whether I'm getting it or not. Sometimes we get negative reports as well. Knowing the fact that the patient has symptoms, it means something has happened, but the test is not able to capture it up. So those things are like, I really take it seriously that I'm doing my job fulfillingly, right? I cannot miss things from my end because if I'm missing, it means the report will go negative. So for the patient, it will be like nothing has happened or nothing has been picked up on the NGS test.
Correct.
So yeah.
I'll tell you one very small story which will connect with you. Is Elizabeth Blackburn. She has written a book which is my favorite book about the telomeres, right?
Okay.
And it's about longevity. And she has given one live podcast on YouTube. Hardly 22,000 people attended. She's a Nobel Prize winner. Okay. And if you look at any famous influencer, there will be millions and millions of people who will be visiting their live. So science is not fancy, but science is so fascinating, right? And when you were explaining that I look at the symptoms and I look at the findings and I try to correlate, that's a purely a researcher's mind, right? That's a pure scientist's mind, which you do not need external excitement. That search itself excites you.
Yes, I understand that because I've been doing it every day.
So beautiful.
Yeah. Because to get the details and to search and at the end of the day, if I have got the variant and I know, okay, these are the symptoms and this is the mutation which I have got and it is like perfectly matching. So at least the patient got their diagnosis confirmed that this is what has happened finally.
Correct. But you don't interact with the patients, right?
No.
You
so
don't.
Everybody has their defined role. My role is doing the analysis. If there are any like technical queries, if the patient wants to know on the technical basis what has happened, then I can pitch in. Okay. But in general, we have genetic counselors only who deals with the patients.
So you're typically inside the lab, and you are sitting with your system and with the data and reports and everything. So I was talking to one of the anesthesiologists, and usually the surgeon is the hero, right?
Okay.
And if the surgery is fine, most of the media people and all of us, we go behind the surgeon and we interview them, and even in the newspaper we talk about the surgeon. But the anesthesiologist plays such a big important role. Nobody knows that what he did and how much of the support that anesthesiology created. So the surgeon was performing the surgery peacefully, right? He's taking care of the patient's life actually, and surgeon is operating into this particular thing, right? So similarly, sir is talking to the patients, he's diagnosing, he's counseling, but all that back-end anesthesiologist kind of a work that you do, right?
Yes.
So I think that's pretty exciting.
That is very exciting. That is my motivation to go to office every day and work.
Yeah, I really understand the importance of what you all are doing as a team. And the best part that I have learned in last 1 hour, that we are discussing is each one of you is playing a very specific role, and you are so dedicated to your role, and you are not even realizing that by doing your role perfectly, you're helping the whole team so beautifully. And I'm so happy to see the dynamics between 4 of you. It's really, really good to talk to you, and thank you so much for your time.
Thank you.